Hello everyone, in this video we're gonna see staining method used in histochemistry and cytochemistry. Let's learn some important terms. First of all, histology is the science of tissues, while cytology is the science of cells. Both histologic and cytologic techniques incorporate various histochemical and cytochemical stains, and thus we must know that histochemistry and cytochemistry are very important fields for studying the inner workings of tissues and cells. So let's know more deeply these fields of biology. First of all, cytochemistry. It is the chemistry of cells, and it consists of studying and visualizing the chemical components of cells. To do this, we need some methods. Cytochemistry uses special stains to develop a color for the microscopic detection of cellular constituents such as lipids, carbohydrates, and enzymes. So, cytochemistry needs to use stains to have colors that we can see under microscope in general. It can be light or electron microscope, and with this color we can recognize constituents of the cells. And now let's know about histochemistry. Histochemistry is the chemistry of tissues, and it consists in studying the chemical constituents of a tissue. To do this, also histochemistry needs methods. So, histochemistry uses different stains and indicators which reacts with the cellular components to develop a tiny colored structures that could be easily observed under a microscope. So, we can conclude that both cytochemistry and histochemistry use staining methods to color structures inside tissues or inside cells to be able to observe them under microscope. So we can see that staining is the principal method used in histology or in histochemistry and in cytochemistry, and thus we must understand how we do staining and what is staining. Staining is the process of coloring cells or components of cells or tissues. The goal of staining is visualization by means of coloring. Staining is a sequence of procedures using either natural dyes that adhere to some cells and tissues, or also chemical reactions that result in the formation of precipitates. So we can understand that staining is mainly a way of coloring some parts of tissues or some parts of cells, and this coloration can help us to see these parts under microscope, and this coloration can happen by means of natural dyes or by means of chemical reactions. Someone can ask, what means dyeing? The word dyeing is used when we talk about using a dye for staining. So, if an object is put into a dye solution and takes from it the dye molecule and not the solvent, The process is called dyeing. If, however, the subject takes up the solvent as well, this is called staining. Both processes result in a colored object. Thus, dyeing is then a special way of staining. We can have different types of staining, so we can stain cells and we can stain tissues. For example, when staining cells, we can stain bacteria, and when staining tissues, we can stain the liver. Other types of staining can be in vitro staining, and this is by taking our tissues or our cells in the lab and staining them. In vivo staining is when we are going to stain the tissues inside the organism. Other types of staining we have simple staining and differential staining. In simple staining, we are going to use just one type of stain per slide. But in differential staining, we can use multiple type of stain per slide. Okay, let's see other types of staining. We have a direct staining and indirect staining. So, for direct staining, we are going to use just one stain, and it will be very easy, very simple, because it is done without the help of mordants. And mordants are chemicals that are used to help us colorate some cells. In this case, this procedure is just with few steps done. But in indirect staining, we are going to use mordants. This means that we are going to use some chemicals that will help us to stain some constituents of the cells or of the tissues that will not be able to be stained in case we do not use these chemicals or these mordants. This happens when we use the procedure of dehydration, in which we use some chemicals that are fixatives that help us to stain the cells. So, in this case of indirect staining, it is more complicated procedures. Let's now talk about the process of staining. Staining method pass by some procedures that differ according to the type of analysis. Here we list some of them. So, to stain a sample, we can just do a wet amount. In this case, we need slide, we need our organism, we need the stain, we can need also a water. So, it's with a few steps we can stain our organism. For example, the onion. And we will see in the next slide a video took from YouTube that simplify this. So with just what amount we can stain an organism. Or we can do other procedures in other cases of analysis, and these procedures can be done all together or one or more of them to reach staining our sample. So let's set them. Fixation, dehydration, embedding, sectioning, and staining. So here, before staining our sample, we can pass firstly via all these procedures. And when reaching staining after that, we can do a microscopic examination of our cells or of our tissues.
To make a wet mount of onion cells, place a drop of fast blast DNA stain on a microscope slide. Take a small piece of onion. Peel a piece off the inside epidermis of the onion leaf scale that is roughly 1/2 to 1 centimeter squared. Place the piece onto the drop of stain on the slide. Add another drop of Fast Blast DNA stain on top of the piece of onion. Prepare the wet mount by lowering a cover slip over the stain, starting at a 45-degree angle. If a bubble forms under the slide, additional stain may be added along the side of the cover slip.
So let's take an example of how to do staining in histopathology field. First of all, we have to prepare our tissue. So, for this, we must firstly observe it macroscopically and take the different size. Register them, then also we cut a portion from the tissue and those portions are placed in a cassette. The first step of the process is fixation. Fixation refers to the use of chemicals called in this case fixatives. They are used on tissues for the following reasons. Firstly, to halt the progression of various biochemical processes that lead to cell degradation, and this way we will avoid the degradation of the cell. So these fixatives will help us to not allow cells to be degraded. It helps us also to preserve the natural tissue structure and to retain the chemical composition of the tissue. The fixatives are administered in two ways, or through perfusion or through immersion. We can take some example of fixative products. The most famous is formal, or what we call also formalin, and it is solutions of formaldehyde gas dissolved in water. Second step is dehydration. It is done to remove water from the tissues, and this to solidify them and facilitate the cutting of thin sections of slides more thinly for use in light microscope and thick for the electron microscope. Let's take an example of dehydration products. It is the ethanol, and it is a kind of alcohol. Clearing is a step that comes with the dehydration, and it's called also infiltration. The process of dehydration is repeated and is continued through a hydrophobic clearing substance, and that to remove the alcohol previously used. Okay, so here we have some example of cleaning products. We can use xylene, benzol. Embedding is the next step, and it is done to solidify the tissue and facilitate the cutting. The cutting is the next step, and it's also called sectioning. Example of embedding products is paraffin. The next step is sectioning. It is the process of obtaining thin sections of slides, more thinly for use in light microscope and thick for the electron microscope. We have an instrument used, and it is called microtome. So you can see that in histopathologic field, we need to pass by all those procedures to reach at the end staining. And staining here, it is the addition of a dye to highlight abnormalities and improve the contrast between tissues. We have an example of staining product here. It is hematoxylin that colors the nucleus in blue. There is also eosin that colors the nucleus in pink. So we can see that in histopathologic field, staining is mainly used to highlight abnormalities that can exist in the tissue. It is very important to know the goals of histochemistry and cytochemistry. So why we are doing histochemistry and cytochemistry? It is to achieve and understand, first of all, the visualization and localization of tissues and cell structures. We need also to know the presence or absence of certain products of metabolism within cells and tissues. It helps us also to understand the quantitation of the chemical constituents of tissues and cells. And finally, it helps us to understand and to know the structures of the chemical constituents of cells as they occur in life and change under various conditions. You see how much is very important. Histochemistry and cytochemistry, this word is helping us to understand the cells, how they are working, tissues, how they are working, and it's helping more doctors to understand how is the illness going on or how some tumors are growing. I reached the end of my presentation. Thank you very much for your attention and see you soon.